My name is Randy Schekman. I'm a professor of molecular and cell biology at University of California, Berkeley, where I've been on the faculty for 48 years now. I'm pretty much a Californian. I was born in Minnesota, but we moved to California when I was 10. And I've been to all of my schooling, education has been in California, mainly in the University of California. I was, however, a PhD student at Stanford, where I worked with a Nobel laureate by the name of Arthur Kornberg, and learned a lot about how to pursue a complicated biological problem from him and how he focused systematically on dissecting processes using biochemistry. My work at Berkeley over many years has been to study how cells manufacture protein molecules, some of which have to be exported outside of cells. So our cells have chromosomes that hold roughly 23,000 genes. That means that many different protein molecules are made inside of cells, but about 30% of these molecules have to be packaged for export outside of the cell. These are molecules like insulin that is made in the pancreas and secreted into the bloodstream and then goes around and binds to tissues that take up glucose that we produce from digestion. But there are many other, hundreds, thousands of other proteins that have to be exported. So they have to somehow be distinguished from most of the proteins that just stay inside the cell to do the things that cells need to grow and divide. So over 2 billion years of evolution, a process evolved to encapsulate those proteins that have to be exported, separating them from cytoplasmic proteins. And that evolution started with microorganisms in early Earth, even simple organisms like baker's yeast. And we use the same molecules now 2 billion years later to do many other things, not just make insulin, but also to allow nerve cells to communicate with each other through the manufacture and export of chemical neurotransmitters. So when I started my career, this process had been understood superficially because you could look inside of cells using microscopes and see these organelles communicating, but there was very little, in fact no knowledge about the mechanism of this process, the molecules that organize the encapsulation of proteins like insulin to be secreted. And the people who were studying this in the 1970s when I began were cell biologists who used microscopes to look inside of cells and see the path that was taken by molecules like insulin, but the techniques were not well developed to understand how this process was organized. And so I chose in the first years of my career to work on this process in baker's yeast because it was possible to use a simple microorganism to use a powerful tool, genetics, to discover the genes that are required for essential processes. And so my lab was able to isolate many such genes and to understand at a molecular level how they work. And then when the human genome was sequenced, it became clear that the same genes that we had discovered were also in our genomes, and we're doing the same things in the human brain or in the liver or the pancreas. And so the work that we did in yeast had a big impact on understanding a normal, essential physiologic process. And that was the basis of my having won the Nobel Prize. A protein is a stretch of molecules called amino acids. The amino acids are the building blocks of protein molecules. There are 20 different amino acids. Many of them we have to eat in our food. We make many of them, but some of them we have to eat from our food. But they're all essential. And inside of a cell, the instructions for making a protein come from the genes. The genes are kind of the blueprint for making a protein, and when this blueprint is copied in the nucleus of a cell, The instructions are in the form of a molecule called RNA, which goes from the nucleus into the cytoplasm of the cell. And then there are little machines, they're kind of like sewing machines called ribosomes that take the instructions and stitch one amino acid next to another to make long strings of amino acids. And these long strings fold into three-dimensional structures that are unique to each different protein molecule. And these three-dimensional structures are the enzymes, or the structural proteins that are responsible for building cells and catalyzing the chemistry of life. So almost all of that, the instructions are in the form of protein molecules that catalyze chemical reactions. And so understanding what those protein molecules do is what biochemists and now cell biologists seek to do to understand how cells operate, what the logic is of a cell, and what the mechanics are of making a new cell by cell division. For most laureates, you end up doing interviews like this, whereas before that I did some interviews, but not as many. For me, it offered an opportunity to express my views on other subjects, not just about my science, but about other things that affect scientists and that scientists do. And so I took the opportunity of the Nobel Prize to speak out very forcefully about the importance of public higher education. You know, most of the world, most universities are public institutions, and so they rely on government support to survive. But in the United States, there's a very interesting divide between public and private. There are very powerful private universities, which are outstanding, places like Harvard and Stanford, and they are often very wealthy, not always, but very wealthy, because they have people of substantial financial means who go there and who give a lot of money. And so increasingly, over many years, scientists end up at these institutions because there's more money to do science. The year that I won the Nobel Prize, of all the American laureates, I was the only one from a public institution. That didn't used to be the case when I was younger. Berkeley, my institution, had many Nobel laureates in physics and chemistry, many more than places like Stanford at the time. But that has changed dramatically over years, and now Stanford has a lot more. They deserve it, there's nothing wrong with that, but the vast majority of university students in the US go to public institutions. I was myself influenced by outstanding scientists, Nobel laureates, when I was an undergraduate at UCLA. So I felt very strongly that my career at Berkeley was the result of a public investment. And I've remained there in spite of having had offers elsewhere, because I feel strongly about the mission of public higher education. And so the Nobel Prize offered me an opportunity to speak out on that issue, and I don't know that I can have any effect, but wherever I have the opportunity, I speak about public education. The Nobel Prize also offered me an opportunity to speak out about issues that I feel strongly concerning scientific publication. So that's another thing that has changed dramatically in my career. When I was younger, most of the journals that scientists wished to publish in were run by scientific societies, where all of the decisions were made by active scientists, judging their peers. And I appreciated that, but that's changed quite dramatically to the point where now it's largely controlled by commercial concerns. Publishers like Elsevier or Springer Nature, which are privately held or traded publicly, but which rely on a quite substantial profit margin. Elsevier, for instance, which is the major publisher of the scientific literature, is the most profitable corporation in the world, not in terms of absolute money, but in the profit margin. That's because they rely on scientists to do the work of reviewing their papers, and then they charge a lot of money to publish the papers, much more than can be justified on the basis of the cost of of doing the reviewing. And I have, because I've had a role in journals, I was the editor of the Proceedings of the National Academy in the US, and then I started a new journal, eLife, where I was trying to promote the value of having active scientists make all the decisions. But that has changed quite dramatically. And most scholars feel that if they don't publish in these commercial journals, they won't be able to have an impact. And I've taken a very strong position about that. In fact, on the day of the Nobel Prize ceremony in 2013, I issued an editorial that was written in the Guardian, the newspaper, online, decrying the influence of these journals and saying that I would never publish in them again myself. And there was a press conference on BBC4 that morning from Stockholm where I debated one of these editors online on the radio. Yeah. And my position is very controversial. Very few scientists embrace my view, although they privately agree with me, but they are mostly unwilling to risk not having their papers published in these commercial journals. It really has become so deeply embedded in the culture that I think it's exerted a toxic influence on scholarship. And so I speak out about this wherever I have an opportunity. Well, basic research done at universities is the foundation of medical and technological advancement. And so countries like Spain must continue to invest in the university systems to turn out the scientists that become the workforce and the entrepreneurs who build these industries. In the US, for instance, the investment in public education that exploded as a result of the Russian launch of the Soviet launch of the Sputnik missile was enormously influential in building a generation of scientists like me who came up with the promise of having interesting and productive careers as a result of this investment. University of California also at that time exploded. It grew by a factor of 2 when I was growing up and provided middle-class, working-class families with opportunities to educate their children. So every government, large and small, must see the advantage of building that knowledge base and training young scholars who will find opportunities in their home country to remain and build industry. In the US, we've had this advantage for so long that the whole biotechnology industry grew out of the investment of the government in basic science in both public and private universities. Mm-hmm. So wherever I travel, countries large and small, it's absolutely crucial that the government understand that this is a long-term investment. They can't take the short-term view, because if they do, they will lose their best scholars who will go abroad to have other opportunities. I had an experience last year in Turkey. I visited Turkey for a symposium that was sponsored by a private foundation. And on the occasion of this symposium, they had 7 very promising young Turkish scientists who came and talked about their work. They were given various awards. Every one of them was doing their work abroad because there were no opportunities in Turkey to do the kind of work that they could do in the US, in Switzerland, in Germany, in France. And it was tragic to see these people. They're as talented as anybody anywhere in the world, and yet they have to escape to be able to have meaningful careers. They train, they pay for the education of these kids as they come up through the ranks through university, but then they leave because there's nothing for them to keep them there. So it's so important that no country can miss that opportunity. And Spain does take advantage of this, not as much as they should, but we judge the contributions of Spanish scientists at this award activity for the Jaime I prizes And we see that there are a number of really outstanding young scientists doing their work here in Spain. So they have an opportunity. Very often the most successful ones rely on European support, not Spanish government support, but they're competitive and they do well. So obviously the universities here are all public, but that doesn't mean that they can't be hosts for outstanding science. They are. Well, it serves to promote excellence in science and to identify scholars, very often young scholars, who otherwise wouldn't have that recognition. And so it's a great thing. It's a big deal. We don't actually attend the ceremony ourselves. The ceremony is later on. We're here to make the decisions about who is to be rewarded, and eventually I'll meet some of the people that we recognize, but I know that it means a lot to them, because I have met some people who won the prize in the past. There are two now. There are two panels. It used to be a mixed panel, and it was very difficult. I mean, biomedical science and clinical science are really huge subjects of obvious importance, but they're rather distinct in the way that they operate. And so I've argued over a period of years and finally succeeded this year in getting them to have two separate panels. I'm the chair of the committee that will evaluate biomedical more basic science, and then there's another committee that will consider clinical discoveries. And I think it will be much easier to decide one or the other by these two committees. Well, of course it requires resources. The organization had to find more money to support another prize, and they've done so this year. I hope they'll be able to continue. Well, the one thing that doesn't come with a Nobel Prize is a crystal ball. So I'm asked this question all the time, and my answer is you can never tell. You can never tell. I mean, in my favorite example is the discovery that led to the revolution in what's now called genome editing. So there's a buzzword that many people have heard called CRISPR-Cas9, which is the basis of the discovery was work that was done decades ago to understand how bacteria fight off viruses that infect bacteria. So this is a little known fact, but bacterial viruses that only infect bacteria, are the most abundant biological particle on Earth. If you just look in seawater, each milliliter, about 20 drops, each milliliter contains about 1 million bacterial cells, but 10 million bacterial virus particles. And these two have been battling it out for 3 billion years. There's a constant warfare of evolution back and forth. And one of the ways that bacteria have been fighting off viruses is by creating a little piece of nucleic acid, RNA, that recognizes a foreign DNA, in this case a virus, and this piece of RNA matches a sequence in the virus, but it doesn't match the bacterial genome. And this piece of RNA is attached to an enzyme that chews up the DNA to which the RNA attaches. That's called Cas9, that enzyme. Cas9 is a DNA chewing enzyme. And so that way the bacterium can degrade an invading viral genome right away before it starts to make new virus particles. And of course viruses evolve to be different than the RNA, and then the bacteria evolve. So this goes back and forth for 3 billion years. It's boring, but that's how evolution works. Anyway, this was discovered decades ago, and then a colleague of mine, Jennifer Doudna, who won the Nobel Prize, along with Emmanuel Charpentier, figured out how to harness this by making a synthetic piece of RNA that could then be used to help change a human genome eventually to cure genetic diseases. That all was the technology that grew out of these basic science discoveries. And the point is, if I had told you this story 20 years ago, you wouldn't care, because who cares about whether viruses of bacteria, and bacteria fight it out, good for them. I hope they kill each other. But in fact, out of that came this amazing technological development, completely unpredictable. Well, there's already been that kind of technology for a long time. There are procedures for neurosurgical procedures that involve implanting things into the brain. My wife had Parkinson's disease. She died of this now almost 7 years ago. But before she died, as the disease advanced and the movement disorder affected her, they had a device where they could implant bilaterally 2 electrodes into the midbrain to emit a little bit of electrical stimulation that would calm the awkward movements normally associated with progression of Parkinson's. So that kind of wiring in the brain has already been going on actually for quite a long time. Now, there weren't chips implanted into her brain, it was just probes with a little bit of electricity. But it's not surprising that there would be chips that could be used to produce new information or new information pathways in the brain. I mean, there have to be jobs at the end of the day for these people. There has to be an industry. There has to be industry that will eventually employ these people. That's obviously important. But to encourage young people to be interested in science should be very easy. Young kids are naturally scientists. They ask questions. They experiment in a way. They experiment. And this needs to be encouraged. And to me, the way to encourage them is not by, just textbook learning. It's by doing. You only learn science and appreciate it by doing it. And so when I was growing up, one thing that got me involved was an opportunity to do my own science projects. Every year I would mount an effort to do a science project of my own design, of my own, crazy ideas, and put it on display for students and teachers to see. That was enough to turn me on. And I don't know if that's done as much anymore as it used to be. It's a very inexpensive way of getting kids interested in doing science. So to me, it's the doing of the science more than just the learning of it that's so important. And I think that kind of education needs to be encouraged. Where kids have natural curiosity about the world, and if you give them that opportunity to explore it themselves, with some guidance, many will emerge, with crazy ideas like I've had. Well, yes. So one thing about being a scientist that is not obvious to a young person is you have to be willing to take risks. You have to be a gambler in a way to be a scientist, to make discoveries. After all, when you're discovering something new, that means that you're going to make mistakes along the way. And so I think there are people who have that kind of courage, and that needs to be encouraged. It's not the kind of thing that you can just be smart and work hard. You have to actually also take risks. I've seen at my institution there are very bright kids who come in who like biology, and their parents of course encourage them to go in a professional direction, like to be a physician, because if you're smart and you do well on tests and you're willing to work hard, you will eventually succeed and have a comfortable career and be reasonably well off. But a lot of these kids, although they're very smart, they're risk averse. That they want a sure thing. If they want to know that if they work hard, they'll get it. But working hard doesn't necessarily make discoveries. You have to. That's not an obvious thing about being a scientist. So it's if they're kids who are curious and are willing to take risks, then science may be the kind of career that would be meaningful for them. If they're not willing to take risks, then it's not going to be rewarding. Right. Luck is a factor, but luck can be guided in a way. You take risks, but you understand the risks that you're taking, and you prepare yourself for working around the risky elements of it. This is what good mentors are for, to guide people and to not let them go down blind alleys. And I've been very fortunate to have had great mentors that were able to guide me away from stupid mistakes that I might otherwise have made.